Hi, everyone. Today we're going to talk more about the properties of different kinds of biological macromolecules. Specifically, we're going to focus on the structure and function of nucleic acids, proteins, and carbohydrates. So let's start with nucleic acids. So, before we do all three, let's talk about something called directionality. And directionality is basically the key to understanding the structure and function of macromolecules, I think. And that's because even though they look two-dimensional when we're looking at the structures on the page in our textbook or on this PowerPoint presentation, we need to remember that macromolecules are three-dimensional objects that interact with each other. If you looked at your two hands on a piece of paper, they would look pretty much like, if I flipped one hand, they would look pretty much the same. But my hands do have directionality. My left hand, I can't put it exactly on top of my right hand. They have different orientations in three-dimensional space. So let's talk about directionality in nucleic acids. Nucleic acid, as we said in the last video, is a polymer of nucleotides. Here's the 5-carbon sugar. Here's the nitrogenous base, and here's the phosphate group. When you link nucleotides together, the 5' or starting end of the nucleotide always has the phosphate group. And the 3' or the back end of the nucleic acid always has an OH group. And when you're synthesizing new DNA or new RNA and adding new nucleotides, you can only add the new nucleotides to that 3' OH group down at this end. You can't add new ones at the front. You have to add them on the back side of the nucleic acid. Now, in our last video, we talked about how a DNA is anti-parallel. It's a double helix. One strand would have the 5' to 3' end oriented this way. The opposite strand would have it reversed. Adenine always forms hydrogen bonds with thymine. They form 2 hydrogen bonds. Cytosine always forms hydrogen bonds with guanine, and they form 3 hydrogen bonds. Okay, now let's talk about the directionality of proteins. Now, proteins, remember, are made of chains of amino acids. All amino acids have an amino group and a carboxyl group. When you link those together, the amino group is always at the beginning of the polypeptide or protein chain. The carboxylic acid or carboxyl group is always at the end. And when synthesizing proteins, when you want to add new amino acids, you always add them to the carboxyl end at the end of the protein chain. Now, let's go a little bit deeper into the structure of proteins. Proteins have 4 different levels of structure. The first one is called primary structure, and primary structure is simply the sequence of amino acids in the polypeptide chain. You can determine the primary structure simply from the DNA sequence of the gene, right? You just look up the codons in the codon chart, and you know what the primary structure or the amino acid sequence is going to be. Now, once that polypeptide chain, that primary structure is made, that primary structure can start folding in 3-dimensional space. Sometimes it will fold up into a helix, into a spiral. That's called an alpha helix. Sometimes it'll just fold back and forth on itself, kind of like in pleats, kind of like when you make a little paper fan. Those are called beta-pleated sheets. And alpha helices and beta sheets are both different kinds of secondary structure in proteins. So we talked about primary structure is the sequence of amino acids. Secondary structure is how that amino acid chain folds either into a spiral or back and forth into beta sheets. The third level of structure is called, you guessed it, tertiary structure. And tertiary structure is a 3-dimensional folded structure, and it's determined by the interactions between the R groups on the individual amino acids. Remember in our last video, we talked about how some R groups are hydrophobic and some are hydrophilic. Well, when we get to tertiary structure, all those amino acids with hydrophobic R groups are going to want to fold those R groups towards the center of the protein. And then the amino acids that have hydrophilic R groups we're going to fold those R groups towards the exterior of the protein, because remember, cells are on average about 70% water or more. So you want to have those hydrophilic R groups on the surface of the protein and the hydrophobic ones on the inside. So those alpha helixes and beta sheets will fold in a way to try to put as many of the hydrophobic R groups in the middle and hydrophilic R groups on the surface of the protein. Now, all proteins have the three levels of structure we just discussed. All proteins have a primary, secondary, and tertiary structure. Really complex proteins that are made of more than one polypeptide chain, proteins that are made of more than one piece or polypeptide chain or domain, those have a level of structure called quaternary structure. And quaternary structure are the interactions between multiple polypeptide chain. And in this diagram, you see here's a protein that's made of two different polypeptide chains. And you can see a helix here, and it looks like we have a beta pleated sheet here. A good example of a protein with quaternary structure would be hemoglobin. Hemoglobin, which carries oxygen and red blood cells, actually is made of 4 different polypeptide chains interacting together. So it definitely has quaternary structure. Another example is collagen, which is what gives your skin kind of its spring and stretchiness so that your skin can snap back. Collagen is actually made of 3 alpha helixes that are braided together. Okay, and you can see the effect of collagen if you take your hand. I think you guys can see this on your screen. Take your other hand and pinch the skin on the back of your hand and let go. And when you see how quickly it snaps back, you can thank collagen for that. So that's quaternary structure. Now, all 4 levels are important to the final structure and function of the protein. If you mess any of them up, you're going to change the structure of the protein and change its function. And later on, we'll talk about that. That's called denaturing the protein. And some of the things that can denature or change the structure of a protein would be if you change the pH. If you put the protein in too acidic or too basic of environment, it's going to disrupt one or more of the levels of the structure. If you heat it up, get the protein too hot, that's going to disrupt some of the forces holding the protein together. And changing the salt concentration of the environment. Remember, salts are charged. So if you add salt to a protein solution, that can disrupt the function and structure of the protein as well, because those charged ions from the salt will interact with the hydrophilic R groups and the charged R groups and disrupt the structure of that protein. And here's a great summary of the 4 different levels of protein structure. I think you either have this exact same diagram or something very similar in your textbook. Your primary structure is a sequence of amino acids. Your secondary structure is when that chain either spirals into an alpha helix or folds back and forth on itself into a pleated sheet. Those helices and pleated sheets can fold in on each other, forming what are called tertiary structures. That's the way the proteins fold for tertiary structure depends primarily on the R groups, so whether they're hydrophobic or hydrophilic. And very complicated proteins that are made of more than one polypeptide chain have quaternary structure. Now, finally, let's talk about directionality in carbohydrates. Remember, carbohydrates are polymers of sugars. They can form linear chains like you see in this diagram here, or they can form branch chains. And that affects the structure and the function. Both of these carbohydrates, this one is amylose, this one is amylopectin. Both of these are made of polymers of the sugar glucose. Remember, carbohydrates are polymers of sugars. In amylose, those glucose molecules are just linked in a straight chain, okay? And it's very easy to break down or be digested. Excuse me. And amylopectin, there are branched linkages between those glucose monomers, and that gives that a lot more structure. And amylopectin is the kind of thing that would like make fruit jellies gel up. All right, so how these things are arranged and what direction they're arranged in really does affect their structure and function, whether they're nucleic acids, proteins, or carbohydrates. So I hope that all makes sense. Have a great day, and I'll see you next time. Bye.